E. coli, you can observe growth of E. coli, you can observe growth of Enterobacter aerogenes, then you can observe Salmonella and Proteus growth, and you can observe in a large diagram pink colonies and colorless colonies. Pink colonies showing actually lactose fermenting ability of organism. So all lactose fermenter will grow pink in color on Mekong ki agar, or they will alter the color of the media due to change in pH of media. Okay, so no lactose fermenter will not show any color on Mekong ki agar. Okay, so then there are these are actually genera of family Enterobacteriaceae. So around 14 to 15 organisms are included, like Escherichia coli, Klebsiella, Citrobacter, Enterobacter, Proteus, Providentia, Salmonella, Shigella, Serratia, Morganella, Armenia, Edwardisella, Hafnia, and Yersinia. Common pathogens for humans are nowadays are E. coli, Klebsiella, okay, Citrobacter, Enterobacter, Proteus is also common, Salmonella is very common. Then Serratia is actually common in children. Then Yersinia was common in last. If we talk about the historical background of Yersinia, then we can say they are involved in plague causing process. Okay, so here you can observe common diseases and common bacteria. E. coli is involved in urinary tract infections, traveler's diarrhea, and neonatal meningitis. On the other hand, Shigella is involved with the dysentery. So bacterial dysentery, Shigella is the biggest cause of bacterial dysentery, mean to say bloody diarrhea. If patient is reporting with bloody diarrhea with blood, then we can say it's a dysentery. Salmonella is very common in Pakistan nowadays. They involve in typhoid fever and enterocolitis. Okay, Klebsiella is involved. Klebsiella pneumoniae is involved in pneumonia, and Klebsiella oxytoca is involved in UTI infections. Okay, surgical side infections are common with the Klebsiella, and Enterobacter are another cause of pneumonia, UTI, and surgical side infections. Serratia is also common, and Serratia is actually common in people of lower ages. Proteus is very important organism. It causes UTI, bone infection, or they can cause kidney stone in complicated UTI conditions. Yersinia is involved in plaque causing process, enterocolitis, and mesenteric lymphadenitis. Okay, so here are few common species. Here are few common genera of Enterobacteriaceae which are involved in disease. Okay, in humans especially. Then how to differentiate between these genera or different species of these genera? So we have many biochemical. Okay, so we have many biochemicals. We can test every organism biochemically, and then we can identify easily which organism is actually involved in the disease causing process. Okay, so here you can observe indole, methyl red, Vogus Prosker citrate test. There 1, 2, 3, 4 tests are common. We say IM big test. Okay, so if we talk about indole, what is indole? Indole is actually test to check the production of tryptophanase by the organism. If bacteria is producing tryptophanase, then they can ferment tryptophan. Okay, amino acids. So when they when they break tryptophan, then they indole form and pyruvic acid form. So if so then after after a breakdown, we can easily identify indole production by the bacteria. If indole produces, then they on addition of Covec reagent, on addition of special reagent, which is known as Covec reagent, they when bacteria is actually indole producer, then we can find indole as a pink color ring on the surface of broth. Okay, so if bacteria can produce indole, they then they will form pink color ring with the addition of indole Covec reagent. Okay, so it will show the production of indole by the bacteria, and it will due to the presence of tryptophanase. Okay, so all bacteria of Enterobacteriaceae family cannot produce tryptophanase. Okay, so E. coli can produce tryptophanase, and on the other hand, Klebsiella cannot produce indole. Okay, so this is actually very important test to differentiate, especially between lactose fermenter, because E. coli and Klebsiella both are lactose fermenter in Enterobacteriaceae family, and they grow a pink color on the agar. But how to differentiate between E. coli and Klebsiella? First of all, a colonial morphology. Of course, colonial morphology is a very important clue, but E. coli is indole positive and Klebsiella is indole negative, which is another evidence for the correct identification. Then there is another important test which is known as MRVP. So if bacteria can if bacteria achieve acidic pathway, then and they can if bacteria break down the glucose through acidic pathway, then pH of the media, which is actually MR, then pH of the media gets low, and then methyl red indicator become change. Okay, so when color of methyl red indicator change due to pH, they then we can say organism is MR positive. E. coli is a MR positive. On the other hand, if glucose breakdown occurs with neutral pathway of by the bacteria, then acetyl methyl carbonyl produce, which form pink color. Then we say VP positive Klebsiella. So MR, VP are two. Of course, you do not need to study principle, procedure, and method and interpretation of these tests. You just need to know what are actually tests, and what are examples of MR positive and VP positive, and how you can differentiate, how can you differentiate between different genera of Enterobacteriaceae family. So that's why you need to know about the MRVP basics or MRVP or these biochemical tests. Okay, you can observe easily in left-sided diagram, methyl red test, MR test is here. So positive reaction show pink color or red color in the solution. So E. coli is MR positive and E. coli is VP negative. Okay. On the other hand, Klebsiella is MR negative and the VP positive. Okay, so these are actually tests to identify between organisms of Enterobacteriaceae. Another important test is citrate utilization test. What is citrate utilization test? Basic concept or idea behind this test is actually the bacteria, identification of those bacteria which can utilize carbon from citrate. Okay, so we put bacteria on citrate agar. So if bacteria is able to utilize carbon from citrate, then bacteria use carbon as a source of energy from citrate, and pH or pH of media gets changed. So when pH of media change, then pH media changed, then indicator color gets changed. So when indicator color changed, then this show positive reaction or use of carbon from citrate. Then we can say this is a citrate utilization test. Carbon is bacteria is using carbon from citrate. Okay, so every organism from Enterobacteriaceae family is not able to use carbon from citrate as a source of energy. So here are few organisms. You can observe in stress positive reaction. You will observe blue color of agar. Okay, so those organisms which are actually straight positive give blue color reaction or blue color agar or blue color slant. Okay, uh, uh, on agar where negative reaction will not show any color. Okay, positive reaction like Klebsiella and Enterobacter are citrate positive organism. E. coli is citrate negative organism. Here is another important test which is known as urease test. Okay, so UREASE test is actually performed to check the ability of organism to degrade the urea of the organism. If bacteria is producing urease enzyme, they break the urea. When they break the urea into carbon dioxide and ammonia, ठीक है, NH3, and ammonia get hydrolyzed and form ammonium hydroxide, then pH become raised. So when pH gets changed in the media, in the agar which have urea, okay, so we provide urea. A bacteria, if bacteria has urease enzyme, they breaks urea, okay, and then pH, ultimately pH gets changed. So when pH gets changed, then obviously color of agar will be changed. Okay, so positive reaction will appear in pink color. Okay, so if bacteria is producing urease, then they will change the color of urea agar. Okay, and they will show positive reaction in pink color. Okay, so positive reaction is shown by the Klebsiella proteus, and the negative reaction is actually Escherichia coli. Okay, so E. coli is not urease producing organism. They cannot degrade urea, and Klebsiella and proteus are common urease producing organisms, are urease producing organism. Then another important test is triple sugar iron, TSI. Okay, you can understand from the name. There are there are three components. First of all, three sugars. Three sugar means glucose, sucrose, and lactose. Okay, keep in mind, glucose, sucrose, and lactose are three sugars are included in this media with one part of iron. Okay, obviously glucose is very low in this media. Lactose and sucrose are more than glucose with respect to percentage, and iron is also included. There is another component which is a very important, which is phenol red, as used as an indicator. Because if pH in media gets changed, then phenol red will show different color on different pH. That's why we say it's indicator. The phenol red is indicating the change in pH. Okay, so you can observe in right side diagram 1 2 3 4. Okay, so 4 number is actually a normal appearance of this media. Okay, so you can observe slant and butt in the media. Okay, slant is actually a slightly slide appearance of media, and butt is actually a lower part of the of the media. Here you can observe a lower part, and slant is actually a slide appearance of media. Okay, so different reaction are shown on the media. For example, if butt and slants are both are red in color as in first reaction, then you can say and H2S no blackening is present in, then you can say it's actually a slant is alkaline and the butt is also alkaline. So red by red is showing alkaline and no sugar fermentation. Okay, so this is actually a no sugar fermentation. So that's why we say it's a non-fermental. Okay, so non-fermental show red by red appearance, and then yellow by red. If bud is yellow but the slant is red, then we say it's actually, and the no blackening is present, then we say acid by alkaline. Okay, mean to say bud is acidic and the slant is alkaline, and we say it's actually a glucose fermentation. So family Enterobacteriaceae always show glucose fermentation in TSI. If we talk about Acinetobacter, which is actually a cocobacilli, so they usually Acinetobacter bomini is also glucose fermenter. Okay, so those organisms which even cannot ferment the glucose, they will show red by red reaction. Okay, so no lactose, so lactose fermenter, so glucose fermenters and the non-lactose fermenter will appear red. Will appear red by acidic by alkaline. Okay. Then in third reaction, you will observe blackening, presence of blackening with red color slant. This is black color is showing the presence of H2S due to the presence of iron. So those organisms which can produce hydrogen sulfide, sulfur binds with the iron, which is iron is present in the media. So after binding with the iron, sulfur and iron give black color appearance in the media. So those organisms which can produce hydrogen sulfide, they appear usually in this way, and you can observe in the third slide. So H2S producers can also identify through this test. In fourth color reaction, sorry, I have just told you fourth one is normal. No, actually one number is actually a normal appearance of this media. Fourth one is showing a very important reaction, which is actually lactose fermentation. If you find a slant, yellow slant and with yellow bud anywhere in the lab, then you can say it's actually a lactose fermentation. Mean to say bacteria in this slant, in this test tube, is able to ferment complex sugar. Okay, so if you get reaction acidic by acidic, mean yellow by yellow, slant is yellow, bud is yellow, then you can say these are lactose fermenters, and you can say E. coli, Klebsiella, Citrobacter, Enterobacter can show this reaction. Okay, so this is actually a function of triple sugar iron test. Here is a summary of few or family of Enterobacteriaceae. Okay, these are actually characteristics we have already discussed. All are gram negative rods, all are oxidase negative, all are nitrate reductase. Okay, they can grow on Meconium agar. So first four are lactose fermenter and all other are no lactose fermenter. So E. coli gives metallic green sheen on the EMB agar. Okay, keep in mind. Then another things are biochemical identification. So biochemical identification also very important. If you find any reaction, if you find reaction or biochemical reaction according to this table for a specific organ, then you can report. For example, if you are suspecting E. coli, then it means TSI will show acidic by acidic, means to say lactose fermentation. Indole will be positive for E. coli, and MR will be positive. BP will show negative reaction. So and they are also motile. E. coli is motile. Okay, so here are the summary of biochemical identification of different organism family Enterobacteriaceae. Okay, so we will discuss later about E. coli. So if you have any question, you can ask. So let's summarize all these things from start. So here you can observe 8 to 10 very important characteristics of family Enterobacteriaceae. Okay, gram-negative or oxidase-negative, catalase-positive. They can reduce nitrates into nitrates, facultative anaerobe. They cannot form, they cannot produce spores, and they can ferment glucose, and some can produce lactose as well. Sorry, ferment can some and ferment lactose, sugar as well. Okay, they can grow well on monkey agar. So here, here is a very important thing about this family. This is actually a classification of family Enterobacteriaceae, lactose fermenter and non-lactose fermenter on the ability of